Okay. Today we'll talk about the types of biotechnology, the broad classification of biotechnology. So biotechnology, the first type of biotechnology is called red biotechnology or medical biotechnology. So actually from the name, it implies that it is a biotechnology that deals with the making of drugs, making pharmaceutical compounds, making enzymes, making antibiotics, vaccines, and these all can be used as well for molecular diagnostic, and that is the red biotechnology or medical biotechnology. The second type is white biotechnology, and white biotechnology or industrial biotechnology. And it deals, of course, from the name as well, it deals with the industrial and other production process. The third type is green biotechnology or agriculture biotechnology. And of course, from the name, it deals with all the agriculture process like the production of different species of genetically engineered species of food, for example. Okay. The fourth type is the blue biotechnology or marine biotechnology. And this deals with all the products, all the marine organisms that can be genetically engineered for application or for getting a better organism. And the famous example of that is one which we have discussed last lecture, is the Casper zebrafish. Okay. So we have sub application for these four major classes, which include microbial, agriculture, animal forensic, bioremediation, aquatic, medical, regulatory biotechnology. It's all subdivisions of the major red and white and the green and the blue biotechnology. So let's take some general information about each type of biotechnology. So for the microbial biotechnology, actually you manipulate the microorganism, and majorly it is yeast and bacteria. You can create better enzymes using these animals. Using these organisms, because animals probably is not the correct term. You can use these organisms to do efficient decontamination for industrial waste product, consume and break a lot of environmental pollutants. And I have already students doing master, I already now supervise students doing master and PhD studies that can use biotechnology, and we are trying to bioremediate from the plastic. As well, microbial biotechnology can be used to clone and produce important proteins used in human medicine. For the agriculture biotechnology, the United Nations of the Food and Agriculture Organization predicts that by 2050, we will need to feed a world population of around 9.1 billion. And this, of course, requires raising food production by 70%, okay? 70% more than which is already present now. So GM, which is genetically modified plants, are more environmentally friendly and yield more per acre. And resistant, you can provide by agriculture biotechnology plants that are resistant to disease and insects. Okay, so as well, you can provide foods with higher protein or vitamin content. And the famous food which is already in the market is golden rice. And for example, how can you develop a plant that can resist disease or insects using a biotechnology? Let's talk about one example. So naturally, there is a plant which is not used as a crop, as a major crop, and they can secrete a substance that can repel insects. But this plant, normally humans don't eat this plant, okay? But there is another plant, for instance, like maize, for something like rice, for something, for example, and human consume these kind of crops. So by biotechnology, you can isolate the gene from that plant which secrete the toxic material to insect, and genetically engineer the plant, the crop that is consumed by human, to secrete this substance. So now you will have, like, for example, like maize that secrete a repellent substance against the insect, and that will be a genetically modified crop. So you can develop as well drugs developed and grown as plant products. And these better plants ultimately reduce production cost to help feed the growing world population. For the animal biotechnology, animals, of course, is a source of a lot of medically valuable protein, especially antibodies. Animals are important model in basic research. These animals can be used in knockout experiments, and you can design and test a lot of drugs and genetic therapies. The animal cloning is a source of organ transplant. So they do animal cloning and collect organs, and they can do transplant for human. You can do transgenic animal. So what's the transgenic animal? It's a way that you introduce a specific gene inside an animal. So it's a way to achieve large scale production of therapeutic proteins from animals for the use in humans. So for example, introducing a gene of important protein that is beneficial to a human into animal like mice, and you let this animal produce that important protein or enzyme for you. So female transgenic animals express therapeutic proteins in milk. So this is one of the famous example. So human, in this example, the human genes, and actually it's another example, the human genes coding for our clotting protein, can be introduced into female goats for production of this protein in their milk, okay? While gene knockout, you can knock out gene, this means neutralize a specific gene. It's a way to disrupt the gene in the animal and then look at what function are affected in the animal as a result of the loss of that gene. So it's a way to understand the function of the gene if it's already unknown. This allows researchers to determine the role and the function of the gene. As we said, the knockout of the gene will allow a researcher to determine the role and the function of the gene. Since the human are very similar in the genetic complement in the genome to rats and mice, so the gene knockout studies in rats and mice can lead to better understanding of gene function in human. So the aim here, instead of which actually forbidden to do gene knockout in human, and you need to know to understand the function of a specific gene in human. So since you can do it in humans, and the gene is conserved and is actually the same nucleotide sequence in mice or rat, and you need to check that before doing the experiment. So you can do the knockout in the mice or the rat or any model organism that have the gene with the same nucleotide sequence. In these model animals, you can investigate the function of the gene and you relate that to the function in the human. So let's talk about the famous example of the animal biotechnology. And the famous examples comes from initially from Dolly, if you heard about it, and if you heard about Dolly. And Dolly was actually the first mammal produced by nuclear transfer cloning. So Dolly was created by cloning technologies. So it's under the category of biotechnology. It's a promising new technique for improving livestock. So this cloning commercially, if you clone commercially valuable animals, you can use it for human transplantation. But unfortunately, Dolly developed early complication and was killed in February 2003. So that was the story of Dolly, the first cloned animal. So what about forensic biotechnology? Actually, the major technique used in forensic biotechnology is the DNA fingerprinting. And actually, you can by DNA fingerprinting to have the to exclude or include a person from suspicious. As well, you can use it for the paternity cases and the identification as well of human remains. As well, you can monitor the endangered species by DNA fingerprinting and then track and confirm the spread of diseases. So let's see an example here. You can see this actually DNA agarose gel, and you can see from this DNA result from this gel, did the dependent, did the defendant commit this crime? And you can explain based on the gel results. So from the gel result, you can see here, that's the defendant's blood. So they do cuts by using some restriction enzymes. They do cuts in the DNA, so it runs in a specific pattern. So DNA is cut already and runs on the agarose gel in a specific pattern. And then this is the defendant's blood. Then they got some blood samples from the defendant's clothes, one from the jeans, some from the jean, and two samples from his shirt. And this is actually the victim blood. For all these, how many samples now we have? We have 1, 2, 3, 4, 5. One of them from the defendant blood, and these three from the defendant clothes. I mean, that's a blood that have been taken from his clothes, and this actually from the victim blood, actually a sample from the blood. And they do the same cut using the same restriction enzymes or all DNA extracted from these samples. And then you compare the pattern of the cut. So the answer now, there is an evidence that defendant commit a crime. You can see that the position of the band as well as number of the bands on the gel matches with the victim blood. So actually this, you can see here that there are matching between the victim blood and the blood from the defendant clothes. So that's mean that at some point the victim blood have been stained the clothes of the defendant, and there is a complete match not only in the pattern of the cut but in the intensity and the number of the bands that arise due to the cut of the DNA sample that is on the shirt of the defendant. So actually, although the bands are very light for the sample from the genes, they are also match the victim blood. So if you look closely to the blood on the genes, although they are very faint, but you can see that the bands are matching. The bands are matching. So based on this gel, you can say that the defendant is linked to the crime scene. You cannot use this evidence to state that already the defendant have killed or do the murder, okay? It's not a proof that he murdered the victim. It's only linking the defendant to the crime scene, okay? So what about the bioremediation? Actually, you can use biotechnology to process and degrade a variety of natural and man-made substances, particularly those that contribute to environmental pollution. And you can see from the figure how they pour a lot of solutions on a heavily polluted area, trying to degrade these pollutants. generally, the estimated growth of bacteria that degrade components in crude oil is increasing. You can see that there is a major spill that happened in 1989 in Alaska for oil. As well, there are another major spill that occur as well in 2010. And if you have an organism that biotechnologically enhanced or modified that can degrade this oil, you can just have this bacteria with the oil, and it will degrade it into similar products, and it will do its bioremediation. So what about aquatic biotechnology? So the major famous example of aquatic biotechnology is aquaculture. And aquaculture can raising the fish, actually it's raising the fish in controlled condition for the use as a food source, because 50% of all fishes consumed by human worldwide. You can use as well genetic engineers to produce oysters, for example, that is disease resistance, vaccines against viruses that infect salmon and other fin fish. So the transgenic salmon already have been made that overproduce growth hormone. Okay, you can follow this link to see some good examples for the transgenic salmon. For the bioprocessing or bioprospecting, this actually the search for a plant or an animal species, you can get from these species medicinal drugs and other commercially valuable compound can be obtained for human benefit. Marine plankton and the snails found to be a rich source of anti-tumor and anti-cancer molecules. So you can see here, so you can see here how the transgenic mice can be produced, which have already overproducing growth hormone. And you can see there is a major difference between normal salmon and the transgenic salmon. The answer, by creating these transgenic salmon that overproduce growth hormone, it allows the salmon to have fast growth rates over a short period of time. Actually, why is that? To fulfill the requirement for the food for humans, the overproduction of food for humans. So how does this modified salmon help humans? Actually, this transgenic salmon, actually it decreases the time and the expenses required to grow salmon for market sales. Okay, and as well, what about the medical biotechnology? Because it's medical, so it's involved a whole spectrum of human medicine, like the preventive medicine, the diagnosis of human and illnesses, of health and illness, the treatment of human disease, and gene therapy. The stem cell technology, one of the very booming technologies nowadays, it uses stem cells and you can grow in the lab and then treat with different chemicals to allow them to develop into specific kind of tissue needed for transplant. And this is major concern in the scientific field in order to avoid the complication that happen when an organ is transplanted from between two different persons. So they are aiming now to get the stem cells from the same individual and do some genetic engineering and some biotechnological approach on this stem cell to allow it to differentiate into liver, for example, and use this liver and transplant it back to the same individual. And this will minimize or even cancels all the rejection reaction that happen in the process of transplantation. So the current use, the stem cells are used for diabetes and the spinal cord injuries. Actually, genes are headlines in the news always headlines in news. So whatever they explore or discover something that is related to the genes and the human, you'll straight see it in the news, because it's a very the medical biotechnology. It's one of the most important branch of biotechnology that human can be affected by this approach, and as well can get a lot of benefit by this approach. What about regulatory biotechnology? Actually, regulatory, you can summarize regulatory into quality assurance and quality control. So you need, in any branch of the biotechnology we have just talked about, you need to have like a quality assurance and the quality control procedures. So all activities involved in, you need to regulate the final quality of the product. And actually, quality control is a part of QA that involve, you need to do a lot of testing and monitoring of process and application to ensure that the product that you already have made using the biotechnology organism will be the same over a lot of round of experiments, a lot of round of batches. So in other words, if a company wants to make your product in different country, it have to be the same product. So that's why you need a strict quality assurance and quality control to ensure the quality and the consistent product standards. And together, quality assurance and QA and QC ensures that the biotechnology product meet strict standards for purity and performance. So how will medical biotechnology change our life in the years ahead? This actually, the major answer for that came after the discovery, after the finish of the Human Genome Project. Research on function of human genes and factor that regulate genes. That actually was the main aim of the Human Genome Project. While the human proteome, its definition means the collection of all protein that's responsible for the activity in the human cells. So actually, it's the total proteins inside each human cell. While single nucleotide polymorphism, actually it is, which we call it the SNPs, it occurs in DNA sequences, and they are vary between individuals. And these variations are the cause of some genetic disease, for example, like sickle cell anemia. SNPs will help to identify genes involved in medical condition, including a lot of genes, including a lot of disease like heart disease and cancer and diabetes, even behavioral and emotional illness. Examples of SNPs, one of the major SNPs that affect breast cancer, they identify SNP in two of the important genes that control the DNA repair machinery in cells, in breast cells. So they found that, or they identified a number of SNPs in these two genes, which is called BRCA1 and BRCA2, and they evolve in promoting breast cancer, led to the development of better targeted treatment for people who have this specific gene mutation. In another context, scientists have managed to predict who is more susceptible to to a certain type of cancer if this SNP is present in the respective gene. So what are the SNPs? Um, uh, let's see what are the SNPs. So we'll see here example of how we can benefit from the Human Genome Project. So from the figure, based on the figure, you have person one have this nucleotide sequence in his DNA or the gene of interest, while the other two have just one nucleotide have been modified, which is G modified to T, or in another person, G is modified to A. So actually it is the same gene, it is the same gene, but have one nucleotide that have been changed. Scientists found that in these two cases, this gene will generate a normal protein, while the modification of this nucleotide from G to A leads to a protein product from this gene, which is non-functional protein. Okay, so you can understand now just a single change in one nucleotide in a gene, which is called the SNP in this case, can lead to change in the product of the protein of this gene. So the answer, since there can be multiple codons for some amino acid, then even person 2, why person 2 doesn't develop non-functional protein? Because the new codon that arise when translating the messenger RNA of the gene will actually can be translated to the same amino acid. Because if you know that some amino acid have multiple codons, like lysine, for example, have 6 different codons. So any change in one of the nucleotide can be compensated and can result as well into lysine. So in this case, you can understand why this individual gave rise to normal protein, because the change doesn't affect the code for the amino acid. While in this, thus the same protein will be made in both cases. While the type of mutation is called, this type of mutation in this case is called the silent, because it doesn't appear and haven't been translated in the protein and will not affect the amino acid sequence. While person 3 had a SNP that it changed the codon when it's translated, and gave a different amino acid, and this different amino acid was a very dramatic in producing a change in the shape of the protein, leading to give the protein that is a non-function. And of course, this may lead to a disease.